A Carnegie Institution for Science audio press release for November 13, 2014. Switching on a dime, how plants function in shade and light. Photosynthesis is the process by which plants convert energy from the sunlight into chemical energy in the form of sugars. These sugars are used by plants to grow and function, as well as food for animals and humans that eat them. Plants grow in environments where the availability of light fluctuates quickly and drastically, for example, from the shade of clouds passing overhead or of leaves on overhanging trees blowing in the wind. Plants thus have to rapidly adjust photosynthesis to maximize energy capture while preventing excess energy from causing damage. So how do plants prevent these changes in light intensity from affecting their ability to harvest the energy they need to survive? The response has to be extremely swift. A team of researchers led by Carnegie's Ute Armbruster and Martin Jonakas revealed a mechanism by which plants maintain high photosynthetic efficiency in fluctuating light. Their work is published in the journal Nature Communications. The question of how photosynthetic efficiency is maintained affects plants on which humans depend, including crops and even forests, so answering this question has practical implications for improving agricultural productivity. The team, which also includes Carnegie's Ari Cornfield and Joseph Berry, discovered that a protein called KEA3 is crucial for immediate adjustment of photosynthetic efficiency in fluctuating light conditions. Photosynthesis takes place in stages. The first stage absorbs light in the form of photons and uses it to produce energy storage molecules, which are then used to power the second stage, which fixes carbon from the air into carbon-based sugars such as sucrose and starch. KEA3 is concerned with the first stage. Under full sunlight, the energy from excess absorbed photons is intentionally dissipated by the plant as heat. But if the incident light is blocked by a cloud, the plant must switch from dissipating excess photons as heat to harvesting as many photons as possible. Advanced analytic techniques demonstrated that KEA3 acts to accelerate the switch from the full sunlight-adapted mode to the shade-adapted mode. This rapid response to light intensity makes the first stage of photosynthesis more efficient. Mutant plants without functioning KEA3 transporters lost a great deal of harvested light energy as heat during a transition to low light, further underpinning their finding. Jonakas explained that the discovery of this built-in machinery for responding to light conditions makes KEA3 a new target for those interested in improving crop productivity, as well as for advancing basic knowledge of photosynthetic biochemistry. The paper's other co-authors are David Kramer and L. Ruby Carrillo of Michigan State University, Kees Venema of the Consejo Superior Investigaciones Científicas in Spain, and Peter Jans and Lazar Pavlovich of Heinrich Heine Universität in Germany. This image shows an example of how plants face fluctuating light conditions as clouds pass overhead. The photo is courtesy Robert Jinkerson. With the Independence for Nimble Pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what's possible for us, our planet, and our universe.